Hi, good evening. So we'll just wait a little bit longer, okay? Like maybe 2, 3 minutes so that others can join. Then we'll start with the class. Okay, I think let's get started today and people will join as we go. So I'll just share my screen. Okay, so I think that's visible to everyone, right? So, right. So last time, we discussed about mostly the excretory system, though initially we had also talked about um, the lymphatic system. So that was in week 4. So today in week 5, we'll continue with some of the questions left over from last time because anyway, the topics are still within excretory system, so they're quite relevant here. So we'll just continue with those questions after I give a quick sort of summary of what we looked at last time. So this is just to recap. We looked at the lymphatic system briefly last time and we looked at some questions, right? So what what is the lymphatic system? It's essentially a system where the lymph fluid, which is essentially the tissue fluid that filters out of the capillaries, is then taken up within these blind vessels known as lymphatic vessels. So from lymphatic capillaries, then they form lymphatic vessels. and these then drain into larger vessels, which ultimately then uh, uh, circulate the lymph fluid through lymph nodes, which is very essential for the immune system. Right. So, yeah. So we looked at the different types of vessels involved in the lymphatic system, and we looked at the ultrastructure and the structural aspects of the lymph node, which is seen here, where you have the T cell zones, which are in your cortical regions, and you have and paracortical regions, and you have your medulla, and you have the germinal centers of B cells um, over here. And these are the regions where B cells and T cells interact with each other. Okay. So this is what we looked at last time, and then we moved to the excretory system, where essentially we're looking at how blood osmolarity is maintained, how fluids are secreted, and this decision-making process in the kidney that's also regulated by hormones and osmolarity sensing mechanisms in the body, and the major organs being the kidneys, um, and the blood vessels connecting them, as well as the ureter and the urinary bladder and the urethra. So, and we learned about the nephron, which is, you can see here, which are the functional units of the kidney. There are millions in each kidney, and they're the bulk of them. We learned about which part of the nephron is in which region, and we looked at, and now in this class, we look at it in more detail as to what is the molecular composition of these different parts of the nephron and how that contributes to maintaining osmolarity and maintaining the composition of blood and urine produced. So we'll look at that in more detail. So just, for all intents and purposes, we're looking at the journey of the blood and the filtrate that comes from the blood through the nephron, right? Essentially, we are going through the glomerulus and the Bowman's capsule where this ultrafiltration is occurring. So you have the filtrate, which is essentially plasma lacking most proteins that comes out. And then you move through your convoluted tubules. You have your proximal convoluted tubules and your distal convoluted tubules, and in between you have this loop of Henle, the descending and ascending loop. And um, during in the PCT, you have massive reabsorption, so huge amount of reabsorption of glucose, amino acids, sodium ions, bicarbonate, and obviously water. Like majority of the fluids are actually taken in um, at the PCT itself. And then you have the loop of Henle where a lot of this osmolarity maintenance is taking place. And we'll talk about how the thickness and the cellular composition of the ascending versus descending loop of Henle, how that is relevant to maintaining osmolarity, right? So the thickness and the amount of aquaporins are present. And then you have the distal convoluting tubule that also doesn't have much aquaporins. And this is for, fine tuning the ionic composition and the uptake in the body, and a very small amount of ions are actually taken up here. So yeah, and finally it drains into the collecting tubule, which is essentially the final decision maker. So is this the composition of urine fine? Is it dilute? Is it concentrated? Does something else need to be done? And if antidiuretic hormone or vasopressin is secreted from the brain, that actually can act on the collecting tubule and increase water uptake over here. And we'll talk about that later. So yeah, so this is just a glance over what we're gonna discuss and the questions that we're gonna discuss relating to these topics. Okay. Oh, yeah, and of course, this I won't explain again because I went into it in detail last time, but this was essentially the renal corpuscle and all of the cellular sort of morphologies of these different regions of the nephron, um, and what their significance is in the function of the nephron. Okay, so if you want to go into detail for this, it's there in the previous video. Okay, so let's get into some questions now. So I'll put the questions up, and I'm sure by now all of you know you can send the question in the chat and we can discuss it. Yeah, so you can just type in and send like a message, and we can look at the answer there and discuss it and yeah, go into details. Okay, so the first question is, why is the proximal convoluted tubule or PCT highly convoluted rather than just being straight? So what is the logical reason for that? Um, so you guys can answer in the message box if you know the answer. If you're not sure, also, it's totally okay to try. Like, it's not a big deal if it's wrong or something. Anyhow, so just like in the okay. So you know how in the small intestine, in the inner wall of the small intestine, the epithelium is highly specialized, where there are these villi, right? That sort of are like these finger-like projections that come out from the inner wall. So what is the function of those guys, right? It's to increase the surface area, which makes sense, right? If you have a flat surface versus if that flat surface suddenly has many little mountains on it or undulations on it, the effective surface area is increasing. So here also the PCT, the reason that it's convoluted rather than straight is simply that you just within a smaller area pack in a lot more of the tubule, so you effectively increase the surface area for reabsorption. Okay, so the answer here is C option, right? That's just that folding up of the PCT just dramatically increases the surface area, and the more you have surface area, the more you allow these transport, more you have transport proteins and channels and pumps embedded in the membrane, and that enables much more reabsorption of water and sodium and glucose and all of this, since nearly like 70% of the filtrate reabsorption occurs in PCT. This is a very important sort of structural aspect of the PCT. Okay, so the answer here is C. Okay, I hope that's clear now. So we can move to the next one. Okay, which three structural components are consistently present along most tubular regions of the nephron? So these are like like some structures or some type of cells. Um, which one do you guys, if you remember? Does anyone want to try? Okay, I think I'll give this one away and then we can discuss it. So the answer here is A. It's the lumen, which makes sense, right? the tubular epithelial cells, and the peritubular capillaries. So, why is it option A? Because across most nephron segments, the three structural elements that are described in option A are consistently there. Okay, the tubular lumen is what carries the filtrate. Lumen is just the inner portion, right? The epithelial cells actually form the tubule wall, and the peritubular capillary network, it receives the reabsorbed substances from the tubules. So this arrangement is what allows this continuous exchange between the filtrate and the blood, and essentially it forms the basis, right? The anatomical basis of filtration and reabsorption and secretion and all of these functions. Okay. So lumen, tubular epithelial cells, peritubular capillaries, these are very essential for these different functions. So that's the answer. Okay. All right. Okay, let's go to the next question. So in tubular epithelial cells, the membrane directly facing the filtrate is called which of these options? So if you guys remember from, your the diagrams in the coursework, I can go back to that diagram once we have discussed the answer. To anyone who has joined for the first time, you can send the answer in the in-call messages um, message box or chat box, okay? And we can discuss. Okay, so to answer this question, let's go back to that diagram. I think it should be labeled here. Hmm. No, I think unfortunately it's not labeled here. Huh. Is it labeled here? No. Anyway, so the answer is D option. Okay. It's the apical membrane. So why is that? It is the one that faces the tubular lumen and directly interacts with the filtrate. Okay, so these cells are um, the apical membrane, and they contain these transporters, right? Like SGLT, and these move solutes from the urine into the epithelial cells. And the opposite of that, which is the basolateral membrane, that faces the interstitial fluid and the capillaries. And that enables movement of the reabsorbed substances into circulation. Okay, so the apical membrane faces the the lumen of the tubules, and it interacts with the filtrate that's coming into the tubules. Okay, and the basolateral membrane faces the interstitial fluid, so the other side, which allows the movement of whatever is being reabsorbed into the circulation from the filtrate. Okay, so that's that. Sorry, the labeling is not there in this image. Yeah, so I hope that is clear. Okay, let's do another one. So tight junctions between tubular epithelial cells allow passage of which substances most readily? So do you guys remember? If you think back to which part of the nephron is responsible for what kind of absorption, then this might be sort of easier to interpret. Does anyone want to try? So this, it would help to remember what a tight junction is, right? So epithelial cells do have tight junctions between them. These are essentially types of junctions that are that prevent leakage. Okay, so like these junctions try their best to make sure that nothing is getting through. And if something is getting through, it's gonna be really small. Okay. So, if for instance, even in your skin, obviously those are different, and the type of reinforcement in your skin is going to be different from those in your nephrons. But tight junctions in your skin essentially are really, really sort of fail-safe connections that prevent too much leakage of solutes or water. But you will have molecules getting through. So here too, in the tubular epithelial cells in the nephron, these tight junctions keep most things from moving through. So they restrict all of these large molecules like major proteins and stuff, but they allow very small particles like water molecules, sodium and chloride ions. Those pass through different pathways. So this is like a selective permeability that's there, and this supports bulk reabsorption, and it also at the same time stops some very important uh, macromolecules like amino acids and glucose and proteins from being lost during the filtration process, right? So the answer here is B option, which is water and small ions. This is what it allows the passage of most readily while stopping most of the other things like glucose and proteins and larger molecules. Okay. So the right answer here is option B. Okay. So I hope that is now clear. So we can move to the next one. If anyone has any questions or wants me to repeat something or explain something in more details or in a different way, just let me know, okay? All right. The abundance of mitochondria in the PCT, okay, the proximal tubule cells, primarily indicates which of these? So this one is kind of just like more of a cell biology question, but it does talk to the physiology as well. So tell me what you guys think. Anyone? Okay, so the correct answer here is C option, extensive active transport requiring ATP. So why is that? So the PCT actually contains tons of mitochondria because there's a lot of requirement of active transport. So what is active transport? Right? It is just basically passage of some molecule or the other across a membrane, but with the help of a pump that utilizes ATP. Okay, so it's not free diffusion, it's not uh, a gradient based diffusion that just um, where a solute or some particle moves based on a concentration gradient. It is actually, it requires ATP. Okay. So the PCT contains a lot of mitochondria to allow for active transport, particularly via the sodium potassium ATPase pump. Okay, so active transport needs ATP, and a lot of Mitochondrial density would make sure that this continuous energy supply is possible, and allows the movement of substances typically against their electrochemical gradients, meaning that you're moving from a region of low concentration to high concentrations, which is against their concentration gradient. So that's why you need excess ATP. It's like pushing water up something, right? Like if water is flowing down a hill versus if you're pumping water up a hill to like maybe a house there, you will need a lot more energy, right? So this is like that. Active transport is using ATP typically to move some solute or some molecule against its electrochemical gradient. So here the answer, cell biologically also, is just extensive active transport requiring ATP, or option C. Okay. So with that, let's go to the next question. So sodium entry from the tubular lumen, which we were talking about, into the epithelial cells is mainly driven by which of these um, sort of reasons? So if you guys think back to how this is working, and we'll talk about it in a little more detail in some slides after this. Do others also want to try answering? If you guys don't know, just you can type the answer that you think it is into the message box and send it, and we can all discuss. Okay. So here the right answer is option B. Okay. So why is that the case? So you guys have to remember that sodium concentration is typically lower in the cell and higher outside the cell. Okay. Whereas potassium is the other way around, right? Potassium has higher cytosolic concentration in comparison to the extracellular concentration. So typically sodium and potassium have opposite electrochemical gradients. So sodium enters epithelial cells because both its concentration gradient, which is essentially higher outside and lower inside the cell, and its electrical gradient, okay, this favors inward movement of the sodium ions. And the sodium potassium pump, which is on the basal side, it keeps this intracellular sodium low on purpose, and maintaining this gradient, and indirectly it powers many other transport processes. So here it is, uh, yeah, entry from the lumen into the epithelial cells is just driven by electrochemical gradient, which favors sodium influx because cytosolic sodium concentration is lower than extracellular sodium concentration. Okay, so I hope that's clear. Let's move to the next one. Okay, so I mentioned this a little earlier about the um, how the SGLT is found on the apical membrane, which is it faces the tubular lumen, right? And it interacts with the filtrate. So this SGLT represents what type of transport? I was just talking about types of transport, right? So what what does the SGLT do? Okay, so the answer here is that it's secondary active transport. Okay, so let me just explain why that is the case. So basically, the other options here, by the way, simple diffusion, for instance, that's just a type of passive transport where there's no energy used, it's just moving along its concentration gradient, it's not doing anything crazy. Primary active transport, it directly uses ATP, and secondary active transport, it indirectly uses ATP. So how does that work? So first, primary active transport, which is what you guys have heard of, right? Where you're moving some substance against the concentration gradient using ATP directly. Okay, and that means the transporter itself is interacting with and breaking ATP. So ATP is being converted to ADP and phosphate and energy by the transporter itself. Okay, wait, I'll just, yeah, by the transporter itself. Okay, and that energy is what is, maybe if the ATPase has some certain conformation, that energy is what's changing the protein or the transporter conformation and allowing the molecule to be moved across or pushed across the membrane. Right, so one of the classic examples of a primary active transport type of pump is the sodium potassium ATPase, right, that you have heard of. So what it does is that every cycle you have these three sodiums that are pumped out of the cell, whereas two potassiums are pumped in, and it uses one ATP. And in both of these cases, you have um, movement against the gradient, right? Yeah. So, yeah, so that's very important for maintaining an ionic gradient and maintaining like membrane potential and so on. So now what is secondary active transport in comparison to that? So here, this transport is using energy that is stored in an ion gradient that was created by primary active transport. So that sounds a bit funny, right? What does that mean? So it's not using energy directly. It's sort of like jumping off of primary transport. So let me explain. So so I explained how the primary pump works, right? So you have, say, sodium potassium ATPase, and it's pumping sodium out. Intracellular sodium is becoming low or lower, and it's vice versa for potassium. So you have this gradient, and sodium wants to move back into the cell. It wants to move along its concentration gradient, which is from out to in. And this actually creates a type of energy. So the cell exploits this type of energy, and it lets sodium come in through the whatever the active transporter, but it drags some other molecule inside with it. So that's where you have these examples of like symporters, right? Or co-transport. So both substances in this case are moving in the same direction, like your SGLT. So your sodium glucose transporter, which is a type of symporter, essentially works like that. Sodium moves along its concentration gradient. So it moves from high concentration outside the cell to low concentration within the cell, and glucose moves against its gradient, but they move together into the cell. So the energy source is essentially the sodium gradient, and that is helping glucose essentially sort of like hitchhiking, right? It's sort of helping glucose hitchhike its way into the cell. So it's not directly using ATP, it's utilizing that gradient, and that gradient is preserved by primary active transport, which is your sodium potassium pump. Okay, so I hope that's clear. You, along with symporters, you can also have antiporters, which is like an exchanger type of thing, right? Where you have, say, sodium. Again, sodium is moving along its gradient into the cell, but this time instead of someone coming in with it, someone is moving out at the same time. So sodium and hydrogen exchanges, H plus, right? That is something that regulates pH and stuff. And that's a type of antiporter or counter transport mechanism. Okay. So, yeah, I hope that's clear. We can move to the next question. Let me see. Okay. So, all right. After glucose enters the tubular cell, how does it typically move into the bloodstream? So, what is the mechanism that these cells utilize for this to happen? Do you guys know?
Glute transporter way of facilitated.
Yeah, yeah, good job. You're absolutely right. That's the correct answer. This GLUT transporter is what allows this glucose to move. So, essentially, once you have glucose accumulating inside this tubular cell, right, it exits, sorry, towards the blood using these GLUT or GLUT transporters. And there's this process called facilitated diffusion. I'll just explain that. So essentially, these carriers move glucose down its concentration gradient, and this is without ATP. Because SGLT brings glucose into the cell, intracellular levels of it then will rise, and that will naturally drive diffusion outwards. Yeah. So now what is facilitated diffusion, right? So that's, we talked about simple diffusion, we talked about primary and secondary active transport. There's also this process of facilitated diffusion. So this is where molecules move down their concentration gradient through a membrane protein without using ATP, right? So it, while it doesn't use ATP, it does use a certain protein. So it's not just through the membrane, it is through a certain protein like channel or something in the direction of its uh, uh, concentration gradient. So it's never against the gradient. So you can have channel mediated diffusion, like it moves through some kind of channel like a ligand gated channel, ion channel, or something, which is essentially like a pore that when some particular ligand binds, it allows um, certain ions to move in or out. So we've talked a lot about these kind of channels before, even in the context of muscular contractions and stuff like that. There's also carrier mediated facilitated diffusion is what we were discussing here, which is where the protein, it binds some certain molecule that changes its shape, and that is what releases it onto the other side. So these GLUT transporters are an example of this carrier mediated facilitated diffusion. So you have this protein embedded in the membrane, that's your transporter. It's in a certain conformation, and then you have glucose that comes in, it binds along with this carrier protein that changes its shape, and that's what allows it to sort of flip and move glucose through. Yeah, so this protein flipping you guys may have studied in biochemistry in your colleges and stuff. So that's essentially what's happening. So you have a lot of glucose outside, very low glucose inside. This transporter, GLUT transporter, binds, and then there's this change, and then it flips over, and it carries the glucose into the cell. Okay, and it's very important in the kidney. While SGLT is pulling glucose into the cell with the help of sodium. So there's this import mechanism. That's one way in which glucose is being reabsorbed, right? Using secondary active transport, which we talked about here. The other way is using this carrier facilitated diffusion. where glucose transporter allow the GLUT transporter allows glucose out into the blood. Okay, so one is pulling it into the cell, and the next it's pushing it out into the blood. So this is what is helping the glucose go from the filtrate into the blood. Okay, so these two mechanisms are very important in the kidney in the glucose reabsorption cascade. Okay, so yeah, let's do another question. So why is it that water follows sodium during reabsorption in the proximal tubules? I think in the last one or two classes, I would have bored you to tears saying this one phrase again and again, that where sodium goes, water goes, where salt goes, water goes. Why is that the case, particularly in the PCT? Anyone want to try? Okay, so in interest of time, if
Sodium increases
uh-huh.
osmolarity, creating an osmotic gradient, I think, ma'am.
Yeah, yeah, you're right. You're absolutely right. Good job. So it is D option, which is sodium increases osmolarity, creating an osmotic gradient. So that's great that you got it. So I'll just explain why this is the correct answer. So when sodium is like reabsorbed, right, it increases the osmolarity in the interstitial fluid, right? And then water is just going to move based on osmotic gradient passively, right? So water follows passively via this osmosis to equalize solute concentrations. So this coupling is why sodium um, essentially is so central to the fluid balance. So wherever sodium goes, usually water will follow, because sodium movement creates osmolarity, that creates an osmotic gradient, and water will just willingly move along that gradient to try to equalize two solutions. You guys would have read about that experiment with the semi-permeable membrane and two like maybe like within one it was like two, like say, bottles, and then they're connected at one part by a semi-permeable membrane, and then you have two solutions of different osmolarity, and then you'll see that water sort of moves across this membrane along its osmotic gradient, so from the more dilute solution to the more concentrated solution until there is an equalizing. So that's exactly what's happening here as well. Okay, so where sodium goes, water is forced to follow because of the osmotic gradient that's created by its movement. So if you use active transport or facilitated diffusion or whatever it is that you're doing to move sodium, then water will also move accordingly. Okay, so great job. I think we have three more questions, then I will explain a few more concepts so that we can tie it all together. Okay. All right, so some proteins that escape glomerular filtration are reclaimed in the proximal tubule mainly through a certain process. So, do you guys know which one? It was, I think, explained in the coursework. Okay, so in interest of time, I'll just tell you guys. Right, so the answer here is C option, which is receptor mediated penocytosis followed by lysosomal digestion. So, why is that the case? So, sometimes what will happen is that some small amount of protein can cross the filtration barrier, right? So typically, what did we see, right? That because of those tight junctions between the tubular epithelial cells, you don't have too much proteins or amino acids and stuff that is just randomly diffusing through or escaping, right? It's just usually water and ions. But sometimes some small amount of protein might cross this barrier. And so you have some specialized receptors on the apical membranes on the side facing the filtrate that bind these proteins and internalize them through this process called pinocytosis. So, have you guys read about phagocytosis? Right? You may have heard about it in the context of macrophages or microglial cells, a type of macrophage, or like these type of cells that are called professional phagocytes, essentially cells that like to gobble up stuff, right? So phagocytosis is a process by which it's an cytoskeletal dependent process by which certain cells gobble up bits of apoptotic debris, some solid parts of cells that were some exosomes or some pieces of cells. Maybe the cell died and then it split into many pieces, and then phagocytes can take up these pieces, or they can take up entire cells or bacteria or some sort of threat in the body, right? So pinocytosis is another sort of form of that, but here it's more liquid cargo. Okay. So it doesn't take up debris and solid pieces of cells and stuff, but it can take up parts of solutes. Okay. So this yeah, so these receptors allow the cells to be pinocytosed, um, these proteins to be taken into the cell by pinocytosis, and then you have lysosomes. So you guys know about lysosomes, right? These degradative uh, little bags in the cell. They're called suicide bags, right? You guys have heard of them. So they are um, yeah, so then they have a lot of these um, proteolytic enzymes, right? That, and they function in this acidic pH that allows them to be activated and degrade different cargo inside the lysosome. So they degrade these proteins that were accidentally sort of escaping filtration. They degrade them into amino acids, which then return into circulation. So even though that protein may not be sort of moving around exactly in the form it was, we're not losing the valuable amino acids, and we're retaining that raw material, which can be used for some other process. So we're retaining the raw material in this way. Okay. So receptor mediated pinocytosis is a specialized process that allows the intake of these proteins in solute as a solute, in the that have escaped from the filtrate, then they're digested, and this amino acids that are resulting from this process are then able to be used in the body. Okay. Okay. All right. Another question. So imagine there's a diabetic patient who begins to excrete glucose in urine. You guys have heard of this phenomenon, right? Where patients who have diabetes mellitus can have glucose in their urine, and this is abnormal, right? You shouldn't have excess glucose in the urine. So you can actually diagnose people or at least send them for more tests if this is an preliminary finding. So what could be the best physiological explanation for this? If you guys think about it, anyone wants to try?
Excess insulin blocks glucose uptake, ma'am.
So, no, I'll explain what the correct answer is, but I'm glad you're trying. Okay. So I'll explain why the option is actually D, okay, which is that the filtered glucose actually exceeds the transport maximum of SGLT proteins. So, uh, yeah, so let me explain. So, right, so you have so with these kind of carrier proteins that move glucose across the membrane, you can actually have something like a transport maximum, which is essentially a saturation point. So when the plasma glucose rises, like for instance in a diabetic patient, what could happen is that the insulin is not in the correct amount, right? Insulin, either it's not released, um, enough is not released, or enough is not produced by the body because of whatever problems. And this lack of insulin is what causes the plasma glucose to rise excessively, particularly after a meal. So then what happens is this filtered glucose actually exceeds the capacity of SGLT transporters. So we talked about SGLT earlier, right? So the sodium glucose symporter, which are the secondary active transport pumps, right? They essentially move glucose, um, and what happens here is that if you have filtered glucose to this extent where you have so much plasma glucose because none of it is being dealt with by the correct amount of insulin, this exceeds this saturation point of these SGLT transporters. So all of these carriers essentially that bind to glucose and move it through, there is a surplus of glucose. There's too much glucose left over, and that is not able to be reabsorbed from the filtrate. And so you have much more glucose than the number of SGLT transporters that are able to do this job. That's why you have glucose left over in the filtrate that then goes in the urine. And that is what this condition is called glycosuria. You guys would have heard of it. It's essentially when there is um, a bunch of glucose in the urine, and it's a classic sign of diabetes. Okay. So too much glucose, not enough of it, not enough receptors are only there or transporters are only there for um, moving the glucose through from the filtrate to the whatever tissue fluid. And so that's why a lot of it is remaining, and that is called glycosuria. So that's the explanation here, which is there's just too much glucose. It exceeds the saturation. Okay. All right, I think one last question before we move to discussing some topics. Right, so despite a large sodium reabsorption that's seen in the PCT, in the proximal tubule, its tubular concentration remains nearly constant, right? So there is this mechanism that allows that. So why is that, right? So anyone wants to give it a shot?
Sodium secretion balances absorption.
Hmm. So I can understand why that might be the answer that you come up with, but it's slightly different. Okay, I'll just explain why. So the answer here is actually, I guess, yeah. Huh. So sodium is not secreted. That's the thing, right? Sodium is reabsorbed, and some amount of it is secreted based on if there is a salt imbalance. But typically, it is water that is uh, reabsorbed to maintain osmolarity based on the sodium. So how does this work? So although you have lots of this sodium that's reabsorbed in the PCT, you would think that, oh my God, it's going to become so concentrated with sodium, right? But water is reabsorbed in nearly equal proportions. I was mentioning, right, that if sodium is going through, so is water. So because both the solute and the solvent decrease together, right, solute being sodium and solvent being water, because both of them decrease together, the concentration of sodium in the tubular fluid of the PCT, it remains relatively constant. So this is called isoosmotic reabsorption. Essentially, you have obviously the mechanics of sodium reabsorption and the mechanics of water reabsorption is different, but the point is that sodium going in, lots of like 70% of the sodium going in or more going reabsorbed at the PCT level. Water is also being reabsorbed proportionally. It's like imagine you have like a bowl of water, and then it has, some x ml of water and some salt, and then suddenly you put like a big tablespoon of salt in, right? So that's going to become really concentrated. But to balance it out, you add the equivalent amount of water required to make it the same concentration it was before. So you pour more water into the bowl. This is basically what's happening here. Okay. So that osmotic homeostasis is being maintained because both sodium and water are being reabsorbed in a proportional manner such that the concentration is not drastically changing at any time. Okay, so that is the explanation here. I hope it's clear. I hope it's concise, right? It's not option A here, huh? Because it is that sodium secretion is not really happening here. It's sodium reabsorption more like. Um, it's not like the PCT is reabsorbing sodium also and then secreting sodium also, right? That doesn't really happen in the nephron. Cool. So I hope that's clear. So now we can talk about a few topics that were covered in the coursework in this week's material, right? So one of them is this very important protein called the aquaporin, right? So aquaporin is a type of water channel, and it's a very, 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 very important type of channel protein. It's not found in all types of cells. So they're these small integral membrane proteins. That essentially integral membrane proteins are basically those that are found embedded inside the membrane and are sort of there. They have portions that are there both on the intracellular and extracellular sides, right? Um, and they exist both in the animal and plant kingdoms, right? And mostly they're very similar. So they have these six transmembrane helical segments. So this region in the transmembrane domain would have these helical segments. So you guys remember from protein secondary structures, you can have alpha helices and you can have beta sheets. So there are these helical segments over here and some shorter helical segments that are there in the cytoplasmic and extracellular regions also. And this part, which is very important, is this aqueous pore. So this narrow aqueous pore is there that actually allows the water movement. So the primary function of most of these aquaporins is to transport water across cell membranes. And it's very important to be able to do that because you have this lipid bilayer, right? So this is, inside it's pretty hydrophobic. So you do need to have these kind of channel transporters that can allow the free movement of water. Okay, so this allows water movement in response to osmotic gradients, right? So you have active solute transports like what we discussed earlier with sodium potassium transport and other types of um, different types of solute transports that maintain osmotic gradients via salts, via the movement of salts. And that's what sets up the osmotic gradient of water. So basically, if you have more sodium outside and less sodium inside, that's an osmotic gradient of water already, right? Because with respect to sodium, water's concentration is different on either side of the membrane. So that osmotic gradient responding to that water will move and that it will move through these aquaporins. And because the water transport capacity of aquaporin monomers is quite low, membranes that require them actually have a very high density of aquaporins. You can have up to 10,000 per square micron. right? And to increase water permeability substantially above that in the absence of aquaporins. So aquaporins are not the only transport mechanism, but they substantially increase the ability to move water across a membrane. Okay, so a little more into detail. So typically, as I mentioned earlier, most cells don't really have a lot of these aquaporins, and all biological membranes are reasonably water permeable because you can have water diffusion through membrane lipids regardless of these lipid tails, right? So the cell volume equilibrates quite quickly, even if there is some osmotic gradient created, right? If that didn't happen, what would happen, right? If you have slow aquaporin based transport, and suddenly you have a cell that has been put into a slightly more hypertonic solution, meaning that the solution outside has higher salt, that cell would just shrink, right? Mean, or if it is put into a hypotonic solution, meaning a cell in a solution that has lower salt, that cell would just explode, right? So the cell itself needs to have this equilibration mechanism so water can diffuse and maintain um, osmolarity and isoosmotic tension between the inside and outside. But so then why do we need this, right? Why do we need higher membrane water permeability in some types of cells? So an example is, of course, what we were just now studying, which is the fluid secretion and absorption, which is very important in kidney tubules, also in epithelial cell layers, particularly, of course, in the kidney tubules, also exocrine glands, where normally the kidney function and secretion of bodily fluids, also like the saliva that's secreted, this require high aquaporin facilitated water permeability, trans-epithelial water permeability, basically water moving across the epithelial layer of cells. So this high water permeability facilitates water movements across fluid tissue barriers, right? And that's what allows the secretion, absorption, reabsorption mechanisms like we studied in the kidney tubules. So this is basically we have some homeostatic level of preserving osmotic tension between the inside and outside of a cell. That's just by free diffusion of water across any osmotic gradient through the membrane itself, of course. But to further enhance water permeability, we have aquaporins in specific types of cells, epithelial cell layers in different types of tissues that particularly require it, okay, like the kidney tubules. And I found something interesting here. You can read more about it at this article, right? So when people uh, created these knockout mice, so what is a knockout mouse? I'm sure you guys have heard, right? Knock down, knock out, knock in, right? These are basically mice that have this aquaporin. So there are many aquaporins, but some particular aquaporin in this case they're talking about that was knocked out, meaning that this mouse does not contain that gene. It could be deleted by CRISPR-Cas9 methods, right? And this would not, I think typically it's not done in the whole animal, though it could be it could be based on whether this is an embryonic lethal deletion or not. So it could be done in a tissue specific manner, meaning only in the kidneys you don't have aquaporins, or that mouse doesn't have aquaporins, or the whole body is a germline knockout, meaning every single cell in that body does not have that particular aquaporin gene. Okay. So when they analyzed the phenotypes of these aquaporin knockout mice, they saw that, like, this is when they were initially trying to figure out what an aquaporin really does, right? This anticipated role of aquaporins they saw in transepithelial fluid transport, that they were able to prove because they saw that deleting them caused excess urine output and impaired urinary concentrating ability, right? Meaning that in the kidney you need high water permeability because you need to have this proper fluid reabsorption and proper ability to uptake the correct amount of water or secrete the correct amount of water to have regulated dilution or concentration of urine and regulated uptake and maintenance of osmolarity of blood, right? So high aquaporin dependent water permeability is needed in the kidney, particularly PCT, right, for near isoosmolar fluid absorption. And in the thin descending limb of the loop of Henle, which we'll discuss very soon, and for countercurrent mechanisms, which we'll also discuss in the collecting duct. And these are all important when it comes to water absorption above what is already possible by membranes. Okay, so in these mice that didn't have the gene, they had excessive urine output, meaning that enough reabsorption is not being able to take place in the kidney tubules, so there's too much urine coming out. And of course, the concentration of the urine would also be uh, um, perturbed in these cases because, again, reabsorption is not taking place properly. Okay, so that was sort of how these studies of aquaporins and the importance or the relevance in physiology was determined. And Peter Eger, who received the 2003 Nobel Prize for trying to, discovering and finding out how these aquaporin channels allow water to flow in and out of cells. He shares the award with a neurobiologist here. And he interestingly referred to aquaporins as the plumbing system of cells, which makes perfect sense, right? Because they facilitate the correct movement of water and via this osmosis, and they're very important in generating all these different biological fluids. So we are just talking about urine concentration, but also we saw how they're important for saliva, sweat, tears, humor, CSF, cerebrospinal fluid, essentially, right? Aqueous humor of the eyes. So in all of these glands and secretory cells, you have aquaporins that are facilitating these processes, and they're also found in plants. And they're messed up in some infectious diseases as well, which you can read about. Okay. Another thing I thought I would cover before we moved on to the next batch of questions, because this would probably help you in answering them, is a little recap of what is an NKCC co-transporter, right? The sodium potassium chloride co-transporter. So what is it, right? It's a transport protein that aids in the secondary active transport of sodium, potassium, and chloride. So we talked about what secondary active transport is, right? So it's the process. It does not directly utilize ATP, it indirectly utilizes ATP. Why? Because the primary active transport mechanisms utilize ATP directly, ATP lysis directly, and the energy released from it is what they directly use to set up a concentration gradient. And moving along that concentration gradient, you have symport antiport processes set up, which is the secondary active transport process. Without the initial energy expenditure, this process would not be possible. Okay, so this can be for sodium, potassium, chloride into cells. And in humans, there are two isoforms of this NKCC. You can have NKCC1 and 2, and they have some different genes. Um, and NKCC1 particularly is widely distributed throughout the body and is very, very important, particularly in organs that secrete fluids, right? In contrast, you have NKCC2, which is found specifically in the kidney, where it extracts sodium, potassium, and chloride from the urine for reabsorption into the blood, right? So NKCC2 is the guy that we are dealing with currently because that is the one that is specific to the kidney, right? So NKCC proteins are membrane transport proteins, right? And they transport these guys across the cell membrane, and because they move each solute in the same direction, they are considered as symporters, right? Meaning that along with sodium you have something else coming in, right? So both of the things are moving in the same direction. Antiporters would mean that one solute is moving from in to out and another solute is moving from out to in at the same time using the electrical gradient of one of the solutes. So they maintain electroneutrality by moving two positively charged solutes. So that is sodium and potassium. They are each, right, 1 plus, alongside two parts of a negatively charged solute. So that is chloride, right? So chloride is Cl minus. So two chlorides neutralize one sodium and one potassium each. So the stoichiometry of the transported solutes is one sodium and one potassium versus two chlorides. Interestingly, this I just found in Wikipedia, which is the giant squid axons. And if you want to know what they look like this, kind of gross. But this is a giant squid axon. It's literally so huge. It's like as long as like what, like half your finger or something. So these are an exception, interestingly, where apparently you have two sodiums, one potassium. So the electroneutrality is still maintained here, but just the stoichiometry is different. So you have two sodiums and one potassium and three chlorides instead of one, one, two. Okay. So yeah, so electron neutrality is maintained regardless, and the rate of transport of these ions are regulated by phosphorylation sites, which are present on the NKCC isoform itself. Okay, so that's about that. So I think with that little recap, we can move on to the next set of questions. If you guys are ready, if you guys want to take a break or something, you can let me know. नहीं तो I will move on to the next set of questions. Okay, so we'll just go through some of them. I think I have like quite a lot of them, but we don't need to cover everything. We'll go one at a time, आराम से. Okay, so the first question is the macula densa, which we had discussed when we were looking at the anatomy of the renal corpuscle, is located in which part of the neuron? So if you guys can remember what the macula densa is and where it is.
Early distal convoluted tubule.
Yeah, you are absolutely correct. That's the right answer. So I'm sure everyone heard her. It is the D option, which is early distal convoluted tubule. So just to explain to everyone, what is the macula densa, right? It is a specialized group of cells located right at the beginning of the distal convoluted tubule, where it is in contact with its own glomerulus. So it senses sodium chloride concentration and it participates in like the feedback um, a bit in the tubulo-glomerular feedback sort of mechanisms. It's very important for that. So yeah, it's present in the early distal convoluted tubule. So great job. And let's move to the next one. So which of the following substances is actively secreted by renal tubules and contributes to a very short biological half-life in blood? So do you guys, I think this was mentioned in the coursework.
Albumin, I think so, ma'am.
Hmm. I'll just explain it and then I hope it becomes completely clear. So the correct answer here actually is penicillin. Okay. So let me just tell you why that is. So penicillin is actually actively secreted by the renal tubules into the filtrate. Um, it's actually very efficiently secreted, and that's why its plasma half-life is really short. Albumin is a very important protein in the blood, and it should not be in the, it should not be secreted by the renal tubules, right? Because it's a very large protein, so it should not be found in the urine. You should not have albumin in the urine. Ideally, that could be a sign of proteinuria or something, right? It could be some incorrect reabsorption or permeability of the Bowman's capsule. So albumin should not be in the blood. Okay, sorry, not in the blood. I mean, it should not be in the urine, and it should not be filtered out from the blood into the glomerular filtrate only. And if it is in the glomerular filtrate, there's something wrong. Okay, so albumin is typically, a lot of it is there in the blood and the lymph. Okay, meanwhile penicillin, right, the very well known antibiotic drug that you guys are aware of, uh, that is actually very efficiently secreted by renal tubules into the filtrate. And pharmacologically speaking, right, when people design drugs, they want to know what is the plasma half life, right? Because you want to know how long some foreign chemical that you're introducing in your body for for some effect or the other, for curing a disease or whatever it is, like how long is that going to persist in your body? So the half life, serum half life, or the half life in your tissues, these kind of things are very important parameters that people who study and design drugs and look at the pharmacology of them, right, they look at these parameters in great detail. And it turns out that penicillin has a very short plasma half-life unless some modification is made onto it, which allows it to then be retained for longer. So, what would happen if a drug is excreted out really fast, right? Maybe it just won't have time to work, right? So, but then if something persists for too long, that could cause, some unwanted side effects, toxicities. So, the drug needs to be optimized as much as possible such that it has the right amount of serum half-life or tissue half-life that allows the effect to be there but prevents any toxic buildup or prevents like bystander effects or non-specific effects in the body, right? So looking at uh, secretion at the level of the renal tubules helps one study how long penicillin will be retained and whether there is something that could be done to it that could allow it to stay longer depending on what kind of treatment uh, um, needs to be given, right? Okay, so I hope that's clear and I hope that bit of trivia was useful. Let's go to the next question. So, the discovery of aquaporins, which we discussed about extensively, helped explain which of these mechanisms in the kidney or anywhere actually. Anyone want to try?
Bulk flow of water across membranes.
Yes, you are absolutely right. Others, if you want to also say your answers, you're welcome, or you can send your answers in the message box. That's fine as well. Okay, but great job. The answer is indeed C option, which is bulk flow of water across membranes, right? So I explained this in quite some detail just now, which is how aquaporins help move water across, lots of water across membranes. Aquaporins are even found in RBCs, right? And this is an osmosis based movement. And aquaporins essentially just provide like this pathway for the water to move. So along with water permeability of the membrane, this is increasing that by providing a like a channel or a tunnel through which the water will move. All right. So let's move to question 4, which is aquaporins are present in which nephron segments? Um, yeah, I don't know what according to the transcript is, but anyway. So the correct answer here is C option, which is the PCT and the descending limb. Okay. So aquaporins are present in high number in the PCT and the descending limb, and that makes them highly permeable to water. They're actually not there in the ascending limb much, which is very important for dilution of urine. So one set is concentrating, the other set. So we'll talk about how the ascending and descending limb and their differential level of aquaporins are important for concentration and dilution of the filtrate respectively. And um, yeah, so the PCT and the descending limb have a high number of these aquaporins that make them very, very permeable, and that's why the majority of water uptake and water reabsorption takes place in those regions. Okay, let's move to the next one. So the thick ascending limb of the loop of Henle is called the diluting segment. Why? So I was just talking about that. So does anyone want to try as to why that's the case?
It reabsorb water only.
Hmm. So actually, this dilution and concentration, you have to remember, is with respect to the filtrate. Okay. So if water is being reabsorbed from the filtrate is becoming concentrated. Meanwhile, if salts are being taken up from the filtrate is becoming dilute. Okay. So not the blood unto which it's being taken up, but from the filtrate. With respect to the filtrate, we are saying either it's a diluting segment or a concentrating segment. So in this case, the thick ascending limb of the loop of Henle is actually impermeable to water. Okay, so this option C, it pumps out solutes. What would happen if it pumps out solutes? The filtrate remaining actually becomes less concentrated. It becomes more dilute. But actually, the ascending limb of the loop of Henle does not have much of these aquaporins. So it's actually, I don't think it has any aquaporins. So it does not allow water to move through because the descending limb of the loop has a lot of aquaporins. So what's happening essentially is, imagine this is this loop. So the descending limb has a lot of aquaporins that remove water from the filtrate, reabsorb tons of water from the filtrate. So filtrate has become concentrated. So the descending limb is the concentrating segment. Now the filtrate is interacting with the diluting segment at the same time. So what's happening is that there's no aquaporins here. So no more reabsorption here. Instead, you have a whole bunch of ions that are being or solutes essentially that are being pumped out. So that is becoming diluting, and it's diluting the filtrate. Okay, so that's why it's the diluting segment. So these two things, the descending thin ascending limb with a whole bunch of aquaporins and the thick ascending limb with low number of aquaporins balance the concentration and dilution of the filtrate. I think someone raised their hand. Would you like to ask a question? Please go ahead.
Yes, ma'am.
Yeah.
Please explain one more time.
This one, the fifth question.
Oh, yes, ma'am.
Okay, okay. Sure. Um, so essentially, maybe I can go back to the diagram that will make it easier. Let me see. So, okay, I think over here you can see it. Right. So you can see over here, um, this is the descending limb, right, of the loop of Henle. You can see the arrows marking the movement of the filtrate, right? And here you can see the ascending limb. So you can see how the descending limb and ascending limb have quite some different morphology. right? One here, it's pretty thin, and these actually have a very thin layer of cells, while the ascending limb has a thicker layer of cells. So, what is happening in these two limbs, right? Why is it that they're different from one another? So, the so it turns out that in the descending limb, you actually have a majority of the water reabsorption because it has lots of aquaporins. So, two things are going to happen one after another. So, the thin descending limb has a lot of aquaporins that take up water from the filtrate, right? It's all about reabsorption of what is required. So, in here, you have a lot of reabsorption of water. So, now the filtrate inside, right? What is the filtrate? Essentially, it is the filtrate that came out over here. You have filtration at the level, ultrafiltration at the level of the glomerulus. That filtrate comes into the, moves eventually through the PCT, the proximal convoluted tubule. Here, a lot of reabsorption is taking place because there's a lot of aquaporins. From PCT, now it is moving, the filtrate now, which has a lot of water already reabsorbed, is moving into this thin descending limb of the loop of Henle, which has a lot of aquaporins. Further water is reabsorbed. So, what is happening to the filtrate moving through during this time? Filtrate is becoming concentrated because there is less water present in it now, so more solutes. But that is not the end. Some salts also need to be reabsorbed. So now the body is so clever, it has designed the ascending limb to do that job. So the descending limb will reabsorb water. Ascending limb no longer has any aquaporins. So instead, it has transporters that allow for solutes, ions, right, salts to be taken up from here. So then what is happening to the filtrate during that process? First, it was concentrated, but then we removed a lot of solutes also. So then it is effectively diluting it. So you have these two balancing processes before this filtrate moves further into the distal convoluted tubule, which has also a very low amount of aquaporins and does some fine tuning of solute concentrations and so on, and some secretion of certain extraneous materials until it goes to the collecting tubule. Okay, so I hope it's clear now that there are these two sort of warring things, right? There's one sort of water reabsorption taking place in the descending limb, and a lot of the solute uptake in the ascending limb. And these two things allow for first concentrating the filtrate and then diluting the filtrate. Okay, so I hope that's clear. And move to the
Yeah,
next
ma'am, sure, it's
question.
clear now.
Yeah, okay, fantastic. All right, so I think we finished this question. Okay, we're on this last one, I guess, which is the NKCC transporter that we talked about, right? The symporter in the thick ascending limb, right? What does it transport? So if you just remember the what this short form is for, then you will know the answer.
Option A, I think, ma'am.
Yeah, that's very correct. So this NKCC is the sodium potassium chloride transporter. It's a type of symporter, right? It moves one sodium, one potassium, and versus that, it moves two chlorides also alongside it into the cell. So it is, these charges are neutralized and it's moving it all into the cell along or driven by sodium's electrochemical gradient. Okay, great. So that's that. Let's do some more questions. If anyone has anything else that they want some clarification on, just let me know. Okay. Okay. So, okay. I think I've repeated this question. So the NKCC system is classified as which one? So if you guys remember, you can just quickly tell what type of transport it is, right? If it's not clear, I can just go over it again. But I'm explained about the types of active transport and what it
Primary
means.
active transport.
Ah, yeah. No, no, no. So this one is secondary, okay? This one is secondary active transport because it works in a
Sorry, ma'am. Sorry. Yes, yeah.
That's okay. That's all right. So anyway, now that you've, you know, corrected it, now you'll know forever, hopefully. This one is secondary active transport. Okay, so it doesn't directly utilize ATP. Instead, it utilizes an electrochemical gradient set up by a different pump that is using primary active transport, which is essentially directly lysing ATP. So NKCC is a secondary active transport mechanism and it is a symport type of thing. And in the case of NKCC, it is sodium's electrochemical gradient that all of these ions together are moving along. So that's what's going on here. Okay, let's go to one more question. So this is just sort of a memory question from your coursework. So these loop diuretics that were mentioned, like furosemide, how do they work? Does anyone know how furosemide works? I won't go into too much detail of it, but essentially, how does it? If you know what a diuretic is, then it might be easy to figure out what this compound might be doing. So, do you guys remember what antidiuretic hormone is or vasopressin is? Right. So, what is that? Right. It's a hormone that reduces urine output, makes you pee less, basically, right? So, diuretic is something that makes you urinate more. Tea, coffee, alcohol, these are all diuretics, they make you go to the washroom more. So, loop diuretics, they're just calling it loop diuretics, and that actually makes it easier to figure out what it is. Diuretic is something that,
Inhibiting sodium potassium ATPs.
mm, not entirely. So, it's a good guess, but it's actually option C. Okay.
Okay,
So
ma'am.
diuretic is something that increases urine output, and an example of that is furosemide. And this is what blocks the secondary active transport, in this case NKCC. So it blocks the chloride from binding NKCC, so it doesn't allow the chloride to move through. So what will happen there? So if this loop diuretic binds to the chloride site, so on this NKCC um, mm, mm, channel or transport or symporter, right? There'll be a place where chloride binds, right? That allows the chloride to move through. But if furosemide goes and sits over there, chloride can't bind. And if that doesn't happen, the transporter cannot function because all of these ions, right? The in NKCC, sodium, potassium, chloride, all of them need to go through together. So you stop chloride, you stop the transporter from functioning properly, and this increases sodium and water excretion because if chloride is not going through, then sodium, potassium, none of these are going through. So they are not being reabsorbed. So that is increasing sodium. Like essentially, there's more sodium left over in the filtrate. So more water is left over in this filtrate because where sodium goes, water goes. So you don't have enough sodium uptake, you will not have as much water uptake equivalently. So more sodium and water is excreted. So essentially, by blocking chloride from binding NKCC, you have more sodium and more water being excreted out. So it's making a person urinate more, or it's causing diuresis, right? So that is how this loop diuretic functions. Let's do another question. So I hope that's clear. Okay. Let's do another one. So the physiological importance of the diluting segment. So we just discussed this in quite some detail, right? The diluting segment. This becomes most evident when which of these? So this is like, you can just think about what the diluting segment is doing. So you have to remember what is being diluted over here. Okay. Always remember that when you're talking about diluting segment, concentrating segment, it's with respect to the filtrate. What is the filtrate? It's what ultimately becomes the urine. So regulating the concentration or the dilution of urine, essentially how much of salt or how much of water we are excreting from the body, that is of high physiological importance, because that is what maintains a person's osmolarity, maintains a person's blood pressure, right? All of these things are regulated at, at the level of these nephrons. So, what is the diluting segment doing, right? The diluting segment is making the filtrate more dilute, meaning it is removing the salts or the solutes from the filtrate, but very importantly, it's doing so without water, so it's allowing the formation of dilute urine, but preventing the excessive loss of salts or solutes. So if a person, for instance, ingests a huge amount of water, just decides to drink like liters of water, what will happen? Suddenly there is too much water and this needs to be thrown out of the body. However, if you want to throw out lots of water, you know, sodium might also go with it, because sodium and water are quite synonymous with one another with respect to their movements. So if you drink a huge amount of water, then not only will you lose a lot of water, you'll also lose a lot of sodium. But that should not be the case, right? The body should be able to distinguish that if there is too much water, only the water should go and the sodium should remain, or at least the correct amount of sodium should remain, and not, you should not become low on salts also on top of everything. So the diluting segment is great in this case because it does not have any aquaporins. So salts will not, so salts will not, yeah, so yeah, so wait, diluting segment. Yeah, correct. So salts will move through, but water will not move through. Correct. If there are no aquaporins, your salts will be reabsorbed, but water will not. So in this case, you will have obviously less water reabsorption taking place in the earlier segments, because you will have you will have lot a reduced level of water reabsorption at the level of the proximal convoluted tubule and the descending limb. But the next segment is very critical because that's where you will make sure you don't lose salt, right? So that's just one way of saying that the difference in the level of aquaporins between the concentrating segment and the diluting segment is very, very physiologically relevant. Okay, cool. All right. So again, coming back to this mechanism. So which mechanism ensures that intracellular sodium remains low in the ascending limb, which then drives NKCC function?
Option A, ma'am.
Uh, mm, Actually, no. I'll explain why. So does anyone else want to try what it might be? So it's a good guess, right? But essentially, we want to maintain certain electrochemical gradients across these cells, right? So intracellular sodium needs to remain low, and that is what drives the function of a secondary active transport mechanism like NKCC, right? And the basal um, membrane here that contains the sodium potassium pump. That's what maintains the low intracellular sodium. Because if this gradient is not maintained, how will the NKCC function? If the primary active transport mechanism is not intact, how will the secondary active transport mechanism take place, right? So the basal sodium potassium pump is very important for maintaining the low intracellular sodium, which essentially sets up this electrochemical gradient such that sodium can move across from outside to inside and bring its friends potassium and chloride with it. So the answer here is option B, which is the sodium potassium ATPase on the basal membrane. Aquaporin, you must remember, okay, is there's no active transport in aquaporins. It is just osmosis dependent movement of water molecules. So there is no ATP there, there is no concentration gradient of solutes that the aquaporins are contributing to. Okay, so NKCC essentially I guess the bottom line here is that you have to remember that sodium potassium ATPase are the primary active transport mechanisms that are very important for maintaining an electrochemical gradient of sodium, and using that gradient NKCC functions to then reabsorb um, sodium, potassium, and chloride ions from the filtrate. Okay, so I hope that is clear. And in the thick ascending limb, particularly, that is very important because that is the place where not only do you not have aquaporins, but you have all of the excessive solute up the not excessive, but you have the increased amount of solute uptake that dilutes the filtrate. Okay, cool. All right. So the sodium chloride symport system, or NCC, is, I don't think I have really discussed in too much detail. Where is it located in?
Early distal tubule.
Yeah, yeah. You're absolutely right. It is in the early distal tubule. So this NCC actually replaces the NKCC in the early distal tubule. So here potassium is not uh, involved. It just reabsorbs sodium and chloride in an electroneutral fashion. Okay, so that's what it is. It's a sodium chloride symporter. Um, yeah, cool. Yes, so that's right. Great job. That's the correct answer. Okay. Now I don't know if you guys remember about thiazide diuretics that were, I think, mentioned. So how do they work? I think this one is clear enough if you remember. Okay, so the answer here is option C. Okay, thiazide diuretics inhibit the NCC, the sodium chloride symporter. So that once it blocks NCC, what would happen is more sodium and chloride are not taken up. So you increase sodium and water excretion essentially. So again, the name diuretic should ring a bell here, which is essentially it's making a higher production of urine. Diuretic makes you go to pee more. So thiazide diuretics are going to reduce sodium reuptake or reabsorption, and that will cause more water excretion based on the osmotic gradients. Now, one question that one might ask is, why is it that there is NCC in the ascending limb, but there is, sorry, NKCC in the ascending limb, but NCC in the early DCT, right? Why does it have to change? So I was also thinking that when I was sort of reading up on these things. So why is it needed? It's not like a random thing. It turns out that it's actually a functional sort of shift that occurs. So this thick ascending limb, it's creating this osmotic gradient, but the early DCT is more sort of fine tuning electrolytes. So once the gradient is built, right, this gradient of ions is built, the kidney just doesn't need something as heavy as NKCC transport. So it switches to a sort of more precision like control in the kidney. So I think you can like go into more detail. I'm not really sure about really why, but essentially it's just the potassium at that level is not required anymore because of already there being a very good gradient being set up. So I guess evolutionarily somehow NKCC was no longer needed in those cells and NCC was replaced there. But if someone wants to read more on it, please go ahead. Um, yeah, so now about this thiazide diuretics. So they target these NCC symporters and they block them by binding to them. So essentially stopping sodium and chloride reabsorption. So the sodium stays in the tubule, therefore water will stay in the tubule and more urine is produced. Okay, so yeah, they increase the water excretion, but I think they're not as powerful as those loop diuretics that we talked about. Furosemide, if you remember. Yeah, cool. I think this compound thiazide diuretics, I think they're used in um, patients who have this tendency to get kidney stones, I think. Um, yeah, okay. Anyway, I think, all right, just 10 minutes. Maybe we'll do three more questions and then we can adjourn for today and then um, yeah, we'll continue in the next class. So let's just do these three. So the principal cells and intercalated cells, I think I had talked about these a long time back, so I'll definitely catch you up again with them. Where are they found? Okay, typically, if anyone remembers.
Late distal tubal and collecting duct.
Yes, you will be right about that. Very good. So they are found in option D, which is late distal tubule and the collecting duct. So these cells are really important for regulating the amount of sodium and potassium and the the um, the balance of like acid and base in the final sort of uh, final few steps of urine production and concentration. So it's like the final decision making when it comes to regulating the composition of urine. So these cells like they work together. So the principal cells are important for water and sodium balance, while these intercalated cells, these guys, they are important for the acid base balance. So that's kind of what they do. And yeah, they have some very important transport proteins. I think I have a question on that, so I won't spoil it right now. So okay, so yeah, we have one question here, which is aldosterone. right? So if you guys remember aldosterone, where it's secreted from, and then the renin, angiotensin, aldosterone, RAS system, right? It's very important, right? So it exerts its effects on principal cells by which of these processes? So it's actually there's a impact at the molecular level. I guess I'll just quickly cover it so that we can wrap up the class because it's getting a little late. Which is so the right answer here is option B. Okay, so it's binding these like cytoplasmic mineralocorticoid receptors and then increasing gene transcription. So aldosterone essentially it enters, I was talking about the principal cells, right? So it enters the principal cells. It's aldosterone is like you can see the sterone part, right? So that just essentially tells you that it's a steroid. So it's a steroid hormone. It binds these cytoplasmic receptors and it moves, once it does that, it then moves into the nucleus. So it acts like a transcription factor and it increases the transcription of this thing called ENaC, right? Which is like a epithelial sodium channel and it increases the transcription of sodium potassium ATP. So it is at the level of the gene, it's increasing like transcription, it's increasing the production of the proteins itself. It's increasing the level of ENaC, it's increasing the level of the ATPase. So that's what aldosterone is doing. And now coming to ENaC, which is what I just mentioned. This is located on which membrane of the principal cells? Again, the principal cells we discussed, right? They are controlling the sodium, water, and potassium balance in the latter parts of the nephron. Uh, um, so this ENaC epithelial sodium channel, it's on a particular membrane of these principal cells, and that impacts the function. Right. So the right answer here is, oh, sorry.
Apical membrane.
Yeah. Yeah. It's the apical membrane. I think this got, anyway, you're absolutely right. It's the apical membrane. So ENaC is there on the apical membrane of the principal cells, and it's important for sodium entry from the tubular fluid into the cells. So reabsorption again. Yeah, so ENaC essentially is like it just allows passive movement of sodium from the formed, almost completely formed urine into the cells. There's no ATP. It's like a facilitated diffusion um, type of channel. Okay. So I do have more questions. We'll continue with them in the next class in more detail. I guess because we ran out of time here, we'll also talk about like countercurrent mechanism, which you guys have learned about. Uh, right. So I hope that was useful to all of you, and we'll meet in the next class where we'll continue on these topics and, of course, then also discuss um, the new subject matter as well. So, if anyone has any questions or anything they want to discuss, let me know. I'll be uploading these videos and the course materials and these PowerPoints and stuff into the drive as well.
Yeah, ma'am, sure.
All
Thank
right.
you.
Yeah, great. Good. All right, then. See you guys next class, which is Tuesday, 7 to 9. Okay. All right.